Well, hello everybody, and thank you very much for joining us this morning, or this afternoon, or this evening, depending on where you're calling in from today. My name is Louise Tratt, and on behalf of the British Neuroscience Association, I'd like to give you a warm welcome to this special webinar, Introduction to In Vivo Multi-photon Microscopy, which of course is hosted jointly with our annual supporters, Scientifica. Just before we start, and while people are still joining, we have just a few housekeeping rules to go over. We'll be using the chat and the Q&A function today, so do feel free to say hello and post comments in the chat. But if you have any specific questions for our speaker today, then please make sure you use the Q&A function for these. You should see both the chat and the Q&A buttons at the bottom of your screen. So there will be a Q&A session after the talk today with a chance for you to ask our panelists any questions that you may have. So do feel free to post your questions at any time during the talk and we'll pick them up at the end. You will have the option to ask these questions anonymously if you wish to do so. I'd like to take this opportunity to remind you the webinar is being recorded with the permission of our speaker, and it will be available to watch on demand on the Scientifica website and the BNA's YouTube channel. And finally, just a reminder to please adhere to the BNA Code of Conduct for webinars and engage respectfully online. So without further ado, it gives me great pleasure to hand over to Dr. Zhongmin Liu, who is Product Specialist at Scientifica and will officially start the webinar today.
So let me share the screen now. Hello, everyone. Thank you for the Louis really kind introduction. And I'm Zhongmin Liu. I'm a product specialist from Scientifica. And before joining Scientifica, I was a neuroscientist interested in the in vivo neurocircuit machinery governing learning and motor execution. And hopefully with this webinar, we can convince you, we can help you and empower your research. Like for example, this really nice video captured by one of our customers, Dr. Elizabeth Meyer from University of Bristol, on cerebellum, the calcium activities of a Purkinje cell. You can see our system provides a really nice high spatial and temporal resolution. You can even resolve the nice calcium dynamics in the fine processes of this neuron. And on the right, this is our QR code for the WeChat social media platform. You're welcome to use your WeChat app to just scan this QR code and to join us, connect with us on the social media. And our company is provide the really industry-leading technology and can help scientists to address questions ranging from the level of moleculars to the level of the networks. And we have like a really nice multi-photon imaging system. Here is actually a photo of a Vista scope, can help scientists to study the all the way from the synapses, that is the building block of the neural circuit, and to all the way of the networks, a large scale of networks like here. And we also have a really industry-leading patch clamp phase recording system, which will allow you to do the to study the small local network, and also the to find and discover, explore the molecular properties and molecular machineries governing, like for example, action potential firing, and in the neurons or in other types of cells. We also have the 2P film. This will help you quantitatively address complicated questions in the field of cell biology and as well as in other fields. And we are really grateful to our customers And all the materials, all the examples, really classic, fantastic examples I'm showing today, all from our customers. And we are really glad that we help them and to achieve their own research goals. And our core value is trust. And with all our efforts, and to hopefully we can further empower basic research and medical research all over the world. So the webinar is actually in 4 parts. So the first part, I will do a brief introduction of multiphoton microscopy and to show you, okay, so what is multiphoton microscopy? What kinds of the multi-microscopy technologies now applied in scientists during the research? And hopefully I can convince you or give you actually some flavors to start to understand why multi-microscopy technology is very powerful technology to empower your own research. The second part I will talk about based on some tips to share and discuss with you. And these tips are coming from my own research experience, and as well as the experience from my like top-notch colleagues and friends. And the rest of parts is for more fun. I will show you fantastic classic representative examples using how scientists using multiphoton microscopy to address their complicated biological questions. So two-photon absorption, this idea, this principle was firstly brought up by Dr. Maria Mayer in 1931, actually in her doctoral thesis. And multiphoton imaging is really suitable for deep tissue imaging. I'm going to explain you why. So this is a parallel comparisons between single photon excitation, like confocal microscopy, and multiphoton microscopy, like two-photon microscopy. The major difference is how the way it excites the molecular. In single photon excitation, on the left, it's just use one single photon to bring the electron, excite the electron, and from the ground state to the high energy state, and relax to this state, then emitted photon. And because this is a linear process, the excitation is actually all over places. That's why you have to have a pinhole in your confocal imaging system to arbitrarily reject out of focus fluorescence. On the contrary, two-photon excitation is actually essentially a nonlinear process, which requires two photons almost simultaneously excite this fluorophore. And this is the true point excitation. There's no excitation ongoing above or below this focal point. So to improve to have the maximum signal to noise ratio, the only thing you need to do is to make sure that these PMC, these detectors, can have the maximum efficiency of collecting as many emitted photons as possible. Here, I just want to recap what I was just talking about. Single photon excitation is a linear process. And you can see here when you have the light coming through and it have excitation above and below this focal point. On the contrary, the two-photon excitation is very limited, restricted to this focal point. And even with the zoom in, it's a tiny point. And if you have the scanning go all over this XY plane, you have the true optical sectioning. Another benefit coming from the multiphoton imaging is because it's usually used infrared laser source, ranging from around 700 nanometer to around 1000 nanometer. Compared with usually it's like visible light spectrum used in the single photon imaging. It can, because it's scattering less and it can penetrate deeper. I want to mention here all optical elements of scientific and multiphoton systems currently is coded to optimize transmission from 700 to 1700, all the way to 1700 infrared light. That means we're really protecting your investment. If in the future, with your growth in the research needs, you want to do 3 photon imaging, you don't have to waste a lot of money and resources to get a brand new 3P imaging scope. You just you use your current scientific 2P scope, upgrade to a 3P scope, and you can use the same system you have used for years to do both 2P and 3P imaging. And this, I just want to give you more ideas. Okay, what is the single photon imaging looks like on the right, on the left, and the multiphoton imaging here on the right. You can see, well, by the glance, you will immediately notice that the high signal to noise ratio coming from the multiphoton imaging. Not only you can resolve individual cells here, neurons here, but also you can resolve these fine processes from these neurons. This is almost impossible from this raw data imaging, from the wide field imaging. And also you can see that the background in the multiphoton imaging example here is almost completely dark. And this is kind of really a gloom of the background, high background. So that means multiphoton imaging also have the high contrast. So, and not to mention that like the multiphoton imaging are very ideal for deep tissue and in vivo research. Some audience might already wonder, okay, so why maybe it's saying why, so it seems like the spatial resolution is poor in like the comparing from multiphoton imaging and confocal imaging. Well, so this is the approximation of the formula used for calculation of the lateral resolution. You can see 2 parameters here co-determine the spatial resolution. One is the excitation wavelength. Another is numerical aperture. That means if you want high spatial resolution, like good enough to resolve fine structures like synapses, you can play with this numerical aperture, whereas you can still use 920 nanometer, this long wavelength, for the imaging. And I will further explain in details and talk about Nyquist sampling principles. And hopefully I can convince you then it's not always necessary to push the boundaries of the spatial resolution of a microscope. And I was just talking about the fluorescence multiphoton microscopy. Beyond that, actually, there's more and more applications for label-free multiphoton microscopy. For example, CARS or XRS. This is actually enhanced the Raman process. And SHG, short for second harmonic generation, and also endogenous fluorescence imaging. So this is a really nice picture. It's actually triple color imaging from a tissue mouse kidney. The CARS is used in imaging the lipids, showing in red color. SHG is used for imaging the collagen, as shown in the blue color surrounding this cell. And endogenous fluorescence imaging, elastin, shown in green. So this is really nice, like applications. And also, of course, you can combine fluorescence imaging, and we have like really nice optimized detection units called chromoflex modular, can satisfy your needs to do multicolor imaging up to 4 different colors. And this multicolor imaging actually is, this chromoflex detection modular is inspired by researchers and designed and optimized by Scientifica. And okay, let's again switch our gear to talk about another major application, that is a 2-photon multi-photon flame fluorescence lifetime imaging microscopy. And this is basically to show you the principles of this flame. So flame, so is also, this is again is the 2-photon excitation, how it goes. And the fluorescence lifetime is really a sensitive reporter of the fluorescence in molecular environment. And so we can do, because 2-photon excitation is actually low probability like event, therefore we can do time correlated single photon counting. So this is show how the principle of this time correlated single photon counting, how it works. So the red showing here is actually the laser pulse, and the green here showing here is the emitted photon due to this laser pulse excitation. And you can count all the time from the start to the stop, and have this nice histogram after thousands of times of counting. And you can have the exponential decay fit here. And one of the major flame application is to is actually FRET. So FRET is basically when the two moleculars or two subunits here domains really close to each other, usually within 1 nanometer. This will add another channel of this non-fluorescence, like non-fluorescence, like the energy here, channel here, and therefore the decay time constant is altered. Actually, is going faster in the FRET compared with the normal non-FRET condition. And the flame is insensitive to the changes of the in the excitation intensity, the volume of the sample in focus, or the fluorophore concentration. Well, all these variables you have to consider if you are doing the fluorescence intensity based measurement. Therefore, combining the multi-photon intensity fluorescence imaging and flame, you can have really comprehensive information, quantitative information, help you to address complicated scientific questions at the molecular level. I will further show you with examples, explain to you why this technology is very powerful. And this is just to show you the TP flame system. We offered this integrated solution. So this microscope used for work here. So B is the schematic detailing of the signal flow in a laser scanning microscope system, measuring the intensity. So this, the biggest advantage or benefit is it can really on time and online to capture the details of the dynamics, activity dynamics. And The flame system receives input from the detector, PMT, and also photodiode detecting the laser pulse, right? Remember, we have to do this lifetime measurement. Then this is flame acquisition. Combining these two, we have really comprehensive quantitative of information, what's really going on at molecular level. And so hopefully, before that, I just wrap up a little bit. I'm talking about the most commonly used technology, that is fluorescence multiphoton microscopy. And then I was talking about label-free multiphoton microscopy, some flavors of multicolor imaging, and also another really important application is 2P flame. Here, I just want to show you, give you a little tour of like what's usually a 2P system composed of. So for the current like 2-photon systems, it's composed with four major parts. First, of course, is the laser source. And second part is launch optics, usually contains beam expander, dispersion compensation unit, which is really critical if you are doing in vivo research, and as well as also the AOM or Poco cell for faster laser power modulation. Currently, Scientifica integrate all these parts in the launch optics into integrating in one single box. So, which make the whole system even more reliable and stable for your in vivo imaging acquisition. Now, let's move on to the main microscope body. It contains the scanners, objectives, and the detectors. I just mentioned about the multicolor detectors, chromoflex, modular, and we also have other types of the detectors and data acquisition and going to the computer. Okay, so what's really like unique and nicely provided from Scientifica is we have this relay lens system. So basically, we put relay lens pairs between X scanning mirror and the Y scanning mirror, so that it can be imaged precisely from the X gavel to, for example, here in the Y gavel, and then onto the back aperture of the objective. And this will prevent the wobbling of the expanded beam, and so which basically avoid the weird netting, and this will make the system able to provide high quality imaging all the time, regardless of the scanning angle. And this is also one of the really critical basis why we can further, based on the system, develop the VISTA scope for the large field of view imaging, which I will talk more in details in the future slides. So this is just want to show you the relay lens benefits. And let's take a look at the imaging, this picture at the right. So because all scanning mirrors appropriately image onto each other using the relay lens, that results in really nearly perfectly flat field, the very edges of the imaged area, and also consistent point spread function to the edges. These 5 points, the zoom in of the measurement of the point spread function, you can see both the lateral resolution and the axial resolution are highly consistent across the whole field of view. And so hopefully I convince you or make you start to understand why multi-microscopy is really powerful technology may empower your research. And the second part, I want to share some tips, or actually just bring out some questions you may interested in discuss with me after the presentation. So firstly, is actually the sample preparation, especially for in vivo sample preparation. If you are a neuroscientist who is interested in studying the cortical neural network mechanisms, you probably need to do the craniotomy. Why? That is because the skull is highly scattered tissue. If you are not removing the skull, you pretty much end like this. You almost don't have the efficient focusing. So not to mention like the efficient excitation to begin with. And craniotomy surgeries, as well as other types of surgeries like green lens implantation, requires a lot of practice and patience, and it's really patience. I have some tips to share, and we're also planning for some workshops in the Asia Pacific area. So please talk with us after the presentation with your questions. And another thing I want to bring about is the Nyquist sampling principle. So this Nyquist sampling principle really matters like how do you configure the optimal imaging setup. For example, what kind of frame rate you want to do, right? So the Nyquist sampling rate is a lossless digitization. And this 2 to 3 times of the highest frequency of the signal will give you the optimal efficiency of data collection. What's the problems with the low sampling rate? That is really easy to understand right here. If you have the sampling rate too low, you end up like a straight line instead of this sine wave. So you're really away from like the actual truth. What's wrong with the high sampling? So this is actually oversampling. Well, first of all, the efficiency of data collection acquisition is very low. If you are doing longitudinal line imaging, like 3 hours of imaging every day, you are pretty much going to overwhelm your data acquisition system, data storage system, and also data analysis system, which really a great waste and waste of the time and energy and resources, and also quite unnecessary because you can get pretty much the same amount of information using Nyquist sampling rate. Okay, so that's the talk about the Nyquist sampling principles, how you apply this in your actual application. So I think most of you are really familiar with the calcium imaging, and you have a really great selections of synthetic dyes or like the genetic encoded reporters. Most like famous is the GCAM, right? And the KD value shows the dynamic range, which pretty much you determined before you're doing experiment. And based on like what kind of types of the cells you are imaging, what kind of the neural networks you want to study. Here, I want to talk about the kinetics of these reporters, indicators. Like, let's take a look at the GCAM6S, for example. You can see both rise time, in particularly like decay time constant, is very large. That means if you are determined to use GCAM6S, it's not really necessary for you to do the resonant scanning imaging. You like gobble imaging, like around several hertz of the frame rate, is already good enough. you don't get actual information from the resonance scanning. Of course, if you're considering multi-color imaging, you have to find you have to have the information about the excitation wavelengths, emission wavelengths, all this information, so that you can wisely and also the full advantages of multicolor detection unit I just talked about, chromoflex modular. Now let's have more fun. So here I want to again just express my gratitude to all scientific customers. All these great examples that you kindly provided to us. So this is the layer 5 pyramidal cells in the visual cortex. And you can see this is what really nice volumetric 2P imaging. And you already noticed that the deeper it goes, the more scattering it will be. And the thing is, you can pretty much you can resolve some details on this process in this apical dendrites, but it's almost impossible for resolving any details like spines on this basal dendrite. That is the physics, right? And determining the limitation of the microscopy. There may be some solutions. One I will talk about later on is the 3-photon microscopy, which allow you to go even deeper. And another way is probably using the adaptive optics to minimize the spherical aberrations. And our system, because our system have the Rayleigh lens, so it's very easy for you to add the adaptive optics or other kinds of modulars suitable for your research. And this is to show you the stack of the spines and boutons here. So it's really nice. If you transfect the cells, infect the cells with reporters like GCAM or glue sniffer, you can also do functional imaging using our system. And this is really cool. So this is the zebrafish visual system imaging, and you can see the nice calcium wave propagation, and also the system can help resolve the calcium dynamics in these fine processes. And this is the probably the Drosophila third instar larvae, and that's focusing on this zooming, this is motor neurons, and you can see this motor neurons have really interesting firing patterns here, probably driven by central pattern generator. And okay, so the previous examples are more focusing on the structural imaging applications and in small size animals like Drosophila, zebrafish. And our system actually have a really large space, can have really large space beneath the scope, and and still maintaining the reliability and stability of the scope. And also I want to point out that another two optimized designs we keep in mind with neuroscientists to really make your in vivo imaging on awake behaving animals going up very smoothly. One is acoustic isolated unit. Maybe some of you have this experience, actually including me. The first time I walked into a 2P imaging room, when my colleague turned on the resonance scanner, I immediately noticed a really disturbing noise, also really disturbing my mouse on the rig. And I asked my colleague, what's that? And my colleague told me this is coming from the working resonance mirror. Well, on the contrary, after joining Scientifica, when I visit our customer's lab, and I cannot tell the noise generated by this resonance scanner. It's really quiet, and it dramatically reduces the audible noise. And it's really beneficial for your animals, really shortening the time for habituation, and also beneficial for yourself. And another optimized design is we have this tunable nose piece. We can change the angles of the objective. This can be very useful in several scenarios. For example, one is if you have animals running really specific behavior rig, and you're very reluctant to shoot the animals, which is usually very disturbing. And you can choose, in our system, you can choose to shoot the objective instead. Also, if you're imaging on large animals or some really parietal brain regions, like auditory cortex, you can also shoot the objective to optimize your imaging. And also, if you're doing the green lens imaging, this function will really enable you to maximize the efficiency of the green lens imaging, 2P green lens imaging. So here I want to just give you some really nice examples from our customers. One is the in vivo mouse motor cortex imaging. This mouse is running on the floating board, and with the resonance scanner working, you can see this raw data is almost have no motion artifacts. The imaging quality is really high. And we randomly select some ROIs and use the Fiji ImageJ and do a really simple analysis. And the x-axis is actually frame number. The y-axis is the fluorescence intensity. You can appreciate the very large signal to noise ratio. And this is cool video is kindly provided from Allen Institute for Brain Sciences. And this animal, this mouse, is running on a lateral treadmill. And I think the imaging is done on the visual cortex. The animal is watching this natural scene movie. And you can see even with the objective tilting, we can still get really high quality imaging data at the edges thanks to the relay lens system. And also, this is a very recent studies. Our system can really pave the way to have better understanding of the processing of emotions during sleep, especially the rapid eye movement sleep in humans. And this is the really cool videos from our customers. And they are studying this microcircuit here. You can notice that the dendrites and the somatic activity are changing in the opposite directions. That's why they call it soma-dendritic decoupling during the REM sleep. They also are using the same system to apply optogenetics on to really to determine which neurons which types of inhibitory neurons contributing to such soma-dendritic decoupling. Also, our system can also empower research in the field of neuroimmunology. Microglia is the resident immune cells in the brain. And this really cool video showing that the experimenter from Dr. Longjun Wu's lab at Mayo Clinic put ATP salts here. You can see the processes of this microglia, and also they go like crazy for this ATP salts. Also, with the equipment of the piezo or ETL, you can do really nice volumetric functional imaging across like several focal planes. Here, actually, the recording is volumetric in 8 planes in total. Here just showed 4 focal planes. And you can see with this electric stimulation, different focal planes shows really different activity patterns. Now, let's switch gears to the applications of 2P film. So one of the really great application in examples coming from Dr. Malino's lab in UCSD is it helps scientists to really understand what's really going on this cytoplasmic domain of this kind of the AMD receptors and how this molecular machinery contributes to this specific type of synaptic activity. And another study is actually a development study. This is a non-neuroscience study from Dr. Christopher Anto's lab. And they are using, so here actually is so they have this thread-based potassium reporter. And when they use to do 2P flame imaging, they can have really comprehensive and qualitative information collecting and have a really full understanding how such this potassium leak channel contributes to regulate the morphogen and the growth factor transcription. Also, here I just want to emphasize, like, although multiphoton microscopy is very powerful technology, it cannot help you to address all scientific questions, right? And our system is really versatile and modularized design, and we can combine with other technology. Here, for example, with the electrophysiology recordings to empower cutting-edge biological research, such as synaptic plasticity. So these researchers, they actually do recording spine EPSC calcium transient in the apical dendrites of the dendritic segment of these pyramidal neurons. And also they do pre-synaptic and post-synaptic electrical stimulation and also post-synaptic whole cell recordings of these action potentials to study the Hebbian plasticity in details. By the way, so this combination of the technology can also help address questions on heterosynaptic plasticity and Hebbian plasticity at the same time on the same sample. Also, our 2-photon microscope can be combined with electron microscopy to help scientists build subcellular map of entire brain networks with all the really exquisite details in functional profiles provided by our 2-photon microscope imaging, and also the ultra-structured details as shown in the serial block electron microscopy. You can see this is like imaging on the same spot on the same brain region. So this approach provides a reliable way to overcome the challenging of imaging structures at vastly different scales. Okay, so I hope everyone is enjoying the nice, fantastic examples I just showed you. I'm going to show you and talk and discuss with you about future trends in the multi-photon technology in the last part of my talk. So first question is like, why do we need large field of view? And here we just want to show you with our large field of view multiphoton microscope, aka with the scope, how large you can imagine in the mouse brain. With 16x objective, you pretty much can capture all the information about half of the visual cortex V1 at one time or at once here. If you don't really need to push the boundaries of spatial resolution, you can have a 10x objective and you can have the imaging all over the whole visual cortex in this mouse brain. Or if you are interested in the motor sensory integration or similar questions, you can imaging on this, you can just imaging M1 and S1 all at once. And I want to also tell you my naive understanding. So why do we need large field of view? Well, as we really expand our knowledge how brain functions, we start to realize a lot of cognitive functions, especially complicated cognitive functions such as decision making, always related to the distributed network, not carried on by like a single network, like local network. That's why we want to see as large as possible. Also, we always usually notice that like we are facing really high dimensional data space. And that also requires us to really push the boundaries of field of view in our system. Scientific microscope has the industry-leading field of view as measured in the field number. And this is the actual size we offers from our system. And with 16x objective is 1.8 millimeter in both Gabo scanning or the resonance scanning. With 10x objective, a little bit sacrifice of the resolution, spatial resolution, you will get 2.9 times 2.9 millimeters, which pretty much the size of the field of view a microscope can achieve. But we're selling the microscope at the price tag of a microscope. And also maybe give you more like a straightforward idea how great the system is. You potentially can get the data of more than twofold of increase of a cell number compared with the standard photon microscope. So this is just a picture snapshot of TCAM6S at 920 nanometer, showing you how big of this field of view, 1.8 times 1.8 millimeter. And this is the resonance scanning. Even with the high frame rate, like 30 frames per second, you still get like 1.2 to 1.2 millimeter. With the relay lens system, you get every information, including the edges, like high quality or with high imaging quality. And this is, I just want to show you how powerful of the multi-LI scanning using the RGG resonant Gabo scanning mode. So basically you're doing a screening and you firstly just like do imaging all over this 1.8 times 1.8 millimeter field. Then you notice that some of the subregions have demonstrated very interesting activity pattern. You can have your software to control and it's like do this, like for example, 5 subregions scanning, and this is really cool. And you can still you can resolve the details of calcium dynamics in these fine processes. With 10x objective, some are sacrificing spatial resolution. You can do multi-alloy scanning in this large area, almost 3 millimeter times 3 millimeter, all across like M1, S1, and S2. This is a really cool example from our customer, Dr. Sylvia Schroeder lab. They are imaging on the population activity of retinal axons in most superior platelets. And this is some degrees of gradings. And just focusing on here, the field of view is 1.3 millimeter. And it's really nice because it's really capture all these details of the retinal axon like activities. And we also have really industry-leading 3-photon microscopy solution for you. This I just want to show you 2 examples. One is the volumetric stack imaging with the GCAM stack, which usually have really low basal fluorescence actually, but just show you how nice all of the stack image looks like. And also, this is kindly provided by scientists from Adam Packer lab, University of Oxford. They can capture the neuronal activity deep as like 1 millimeter deep in vivo, which is really, really nice. Also, we provide all optimized solutions for all optical interrogation. This is SLM based holographic photostimulation. And here this really example our scientists captured in Dr. Adam Packer's lab. And we randomly selected a couple of neurons expressing C1, both C1 and GCAM. And I want to, and this traces just show you these selected neurons, how they respond to this holographic photostimulation. But I also want you pay attention to these neighboring neurons. And they also have really interesting response patterns. So when the holographic stimulation simultaneously can stimulate all these neurons. And also you can see these neurons in the neighborhoods light up and have really interesting response patterns. All right, so I hope you really enjoyed the talk and you may be convinced how powerful of this multi-photon microscopy technology. And I'm happy to discuss with you with any questions. Thank you.
Excellent. Thanks so much for that talk there, Zong. That was amazing. I'm sure everyone will agree there were some amazing images and videos, and it was really fascinating to see what you've been doing. So thanks for sharing that with us. I can't see any questions that have come in specifically at the moment. We do have a few more minutes, so please do feel free to post your comments, post some questions while we have Zongmin here at your fingertips, ready to answer any questions that you might have. Elle said that's an amazing talk, so thanks for that. Is there anything else that you have to say today, Zongmin, or any sort of advice other than what you've given in your presentation? If people do want to contact you, if they think of something after the webinar, are you happy to take questions?
Yeah, so we have multiple channels, ways you can connect with us. One is to follow us on WeChat and using this QR code. And also you can contact us with this email address, info@scientifica.uk.com. Or you can also go ahead and sign up for the mailbox for our further news bulletins. And you will receive the latest guides, webinars, and application notes straight to your mailbox. And please look at our learning zone, and we constantly invite neuroscientists to share their tips and experience, and like not just like really restrict to like experiments, right? And also something related to like how to plan your future scientific career paths, and some really deep stuff there.
So that's in the learning zone.
Yeah, and also the NeuroWire, lab hack, all these things. Yeah.
Fantastic. So there's lots of ways that people can keep in touch with Scientifica and also reach out to you too. Maria Louisa has said, amazing presentation. Thanks a lot. So lots of people giving their thanks to you today, Zongmin. Great. Okay. Well, we don't have any questions at the moment. You know, it's quite early here in the UK, so maybe people are still. Oh, here we go. We have a question now. So this is from Jazina. Says, thanks for the presentation. I may have missed this during your talk. What would be a typical frame rate for galvo scanning?
So usually will be around 2 Hertz or like 5 Hertz of this. So typically of this like range. And also that's something to do with the, how to say, the field of the imaging you're going to do, right? So there are like several hertz of frame rates. I actually mentioning about like 512 times 512 pixels. Of course, if you go to like 1024 times 1024, you are expecting even lower frame rate. And if you go in the opposite direction, right, if you have like 256 and 256, you have like a higher frame rate. And also that's really depending on the scanning, how the way of the scanning, right? Actually, so we have like scientists, customers demonstrating that using gobble scanning and like a spiral way. So you actually can skipping some pixels because there is nothing there, right? And they have shown that you can get really high frame rate, like 100 Hertz in the, well, downside is a small size of the field of view, like around 300 microns times 300 microns.
Excellent. Thanks for that. I hope that's answers your question. And just to remind everybody that this webinar has been recorded and it will be available on the website, on Scientifica and BNA's website. So anyone that wants to catch up with this afterwards can do. We have a question now from Gil. Any experience on how long a mouse cranial window can last for in vivo imaging?
Well, so that's really depending. Because I myself I can share with you. Okay, it's really frustrating at the beginning, right? When you're trying to do the craniotomy, it's really a difficult learning curve. But once you're really mastering that, and you're really careful, and also I want to say that also pay great attention for the post-surgical care. You take care of your animals, and you will get great returns. And for my own record, you can pretty much have like really nice high quality cranial window lasting for several months, like over 6 months. And you can still do really nice imaging on spines or boutons from the same cranial window, longitudinally. But the success rate is kind of like depending on your practice, right? We usually say that if you can do such thing, like over 50%, you can have really nice high quality cranial window within a month. You already are expert. Yeah, so I have some tips to share, so please feel free to reach out me. We can talk about, if you don't feel comfortable talk about here, we can talk about that, like discuss about that in private.
Excellent, thank you. I hope that's okay, Gil. If you want to reach out to Zongmin, then you can do that. Okay, so it's 10 o'clock now, so it's the end of our webinar today. Thank you so much again, Zongmin. That's been amazing, and I hope that you have all enjoyed watching today. We'll send round the recording when it's ready, so please do invite your colleagues and peers to have a look. And that's it from us. So thank you very much, and have a great rest of your day or evening, wherever you're calling in from. Thank you again. Bye.
Bye.